Thank you. It's my great pleasure now to introduce to you the keynote speaker, Cynthia Powell. I've known Cindy for a number of years, and she's a great colleague. Cindy started as a genetic counselor and is actually certified as a counselor as well as a pediatrician, cytogeneticist, and a clinical geneticist. She's Associate Professor of Pediatrics at the University of North Carolina in Chapel Hill, and she's the Medical Director of the Cytogenetics Lab there. She's going to talk to us today about the early days on a new U grant that they were funded and Cindy was PI on regarding genomics and newborn screening. So thanks for coming, Cindy.
Thanks, Hans, and thanks to all the organizers for inviting me. It's a pleasure to talk to you today. And what I'd like to do, I have no disclosures to make, and I'd like to give a brief overview of newborn screening criteria, current and proposed conditions screened for in newborns, how next generation massively parallel sequencing may help expand the number of conditions that we could screen for, go over a little bit for those not familiar with that technology, what it is, and how it brings up a lot of ethical challenges if we're going to be doing this, and then to give you an overview of the what are called the NSIGHT projects, the NIH-funded U19 projects, newborn sequencing and genomic medicine and public health, that are studying all of these issues. So I think there's no doubt that newborn screening is successful. This is one of our metabolic camps organized and run by our metabolic dietitians. You'll see Sarika Pendiel in the middle there, and Diane's hiding out in the back, Diane Frazier. But these are children who are teens with inborn errors of metabolism who have been successfully diagnosed early on and treated. Some of the criteria for newborn screening were adapted from Wilson and Youngner criteria in 1968, and those include that there's a treatment available, that early institution of treatment can help prevent the severity of the illness or eliminate the illness, that otherwise routine observation of infants will not allow you to diagnose these conditions, and so a test is required, and that there be a rapid and economical test in order to detect these conditions, which is of good sensitivity and specificity. In 2005, because there was so much variation between states in terms of what conditions were being screened for, HRSA funded through the American College of Medical Genetics a panel that looked at what conditions should be included and screened for in all states. And so they came up with a panel of 29 conditions, now up to 31. That all states should screen for, and that there were also 29 additional secondary conditions that would be detected as part of screening for the core conditions. This committee is now called the Discretionary Advisory Committee on Heritable Disorders in Newborns and Children, and you'll be hearing more later on today from Alex Kemper about the work of that committee. So new conditions can be nominated. But some of the limitations are that there's no screening tool available, that the screening tool is too expensive, and that there's no treatment. If you look at all of the different conditions that were considered by that first committee, there were a number down here, some of which are now included in the RUSP, which at that time didn't have a method of detecting or screening for. And so those that were excluded, there were 23 that were excluded because there was no test available. And 21 of those conditions that were excluded are detectable in some or all cases with molecular genetic analysis. So some of the barriers that have prevented us from being able to screen for more conditions in newborns can be overcome through use of molecular genetic technologies. So a little bit of background about what is massively parallel sequencing or next generation sequencing. What I like to explain to our students and residents is that if you think of traditional sequencing of a gene, that's through what's called Sanger sequencing, and you basically could read all the letters of the alphabet or the nucleotide base pairs in a gene, similar to, let's say, reading one letter that's in a person's mailbox. Whereas next generation sequencing has the potential to look at an entire encyclopedia, a person's entire genome that might be in that mailbox. We can search for mutations theoretically in all 20,000 genes, or you could limit it just to the exome. The whole exome sequencing just looks at the exons. So it's much more limited than whole genome sequencing that would include the exons and introns. But the analysis for this is still extremely complex. We have a long way to go through things like ClinVar. We're learning more about what are significant variants versus what are just benign changes in people's genomes. But we still have quite a ways to go. It's not that different from any other type of molecular genetic analysis. You have to prepare or get your sample, collect your sample, and then prepare the DNA and prepare the library. And then you do the sequencing, which really doesn't take that long nowadays to do the sequencing in a day or so. But you have to do a great deal of informatics on that information with aligning and mapping and variant calling and the analysis. And one thing I want to point out is this is really the part that takes a great deal of time. So even with the best informatics pipeline that may tell you what is likely or possibly a significant variant, humans still have to be involved and sort through all this information, go to databases, go to the literature, look at, is this variant something that's going to cause a problem or not? And so currently that's really one of the things that makes this quite a lengthy procedure, although as I'll mention in a little bit, there are some places that are speeding it up quite a bit. So as I said, with microarrays and Sanger sequencing, we all get variants, right? And that's one of the things we struggle with, in the cytogenetics lab with our microarrays. We're always finding things that, haven't been reported before, but maybe they contain one or more genes, and if there's a duplication, is this something that is going to cause problems? So the variant calling software is important, and then we use a lot of databases. 1000 Genome, now SERGG, is a very helpful database that provides information on variants in a much larger population sample. And then looking at what are the effects of that mutation or variant in terms of what does that amino acid substitution do to the protein product. And then we can also use animal models to look at the significance of a change through use of zebrafish and other model organisms. But there are still a lot of limitations, especially with whole exome sequencing. It's not able to detect trinucleotide repeats, so conditions like Fragile X it can't detect. GC-rich regions of the genome are difficult to analyze, and as we know, not all pathogenic mutations are contained in the exons. Copy number variants are difficult to detect, although that is getting better. Structural variants, the same thing, still difficult to detect, but there are some methods that have been reported where you can find translocations, balanced translocations. And then epigenetic conditions such as some forms of Angelman syndrome, Prader-Willi syndrome that are due to methylation changes you wouldn't be able to pick up. And one of the difficulties, especially in metabolic disorders, is that a lot of genes that code for proteins that are enzymes that or lead to some of the metabolic conditions we're familiar with have pseudogenes. And so whole exome sequencing can pick up variants in these pseudogenes that may look like a pathogenic change where it's really just a change in a pseudogene, so it's not going to impact the protein product. As I said, there's still a long turnaround time for whole exome and genome sequencing. It's still expensive. On a research basis, we budget for about $1,000 per sample. And unfortunately, also for autosomal recessive conditions, you can't differentiate between variants that are in cis versus those in trans. So getting parental samples is needed if you come up with something like that. And then there's the ethical issues. So incidental or secondary findings, and there have been recent papers through the AAP and ACMG and most recently ASHG about concerns about doing this in minors and children because of the issue of incidental or secondary findings. We know that testing can identify variants that are not related to the initial reason for testing. These can include mutations in genes such as those leading to adult onset conditions such as breast cancer. What should be reported back to families? And others have brought up that, well, we know the criteria for traditional newborn screening, but shouldn't we try to expand that definition of treatable? What is treatable, and is it only treatment in the traditional sense in conditions that we should be trying to look for? So for those, for example, conditions associated with intellectual disability, isn't there some benefit to detecting those at an early age before a child would otherwise demonstrate developmental delay, for example? Parents go on this diagnostic odyssey trying to learn what the cause of their child's problems are, and that can be very expensive and time-consuming and frustrating for parents. If you had an answer shortly after birth, you could avoid that diagnostic odyssey, and it would also be cost-effective because you'd have fewer tests done and the expense of that. You could get early intervention services started more quickly in a child and provide the parents with genetic information regarding a potential risk of recurrence, as has been shown, for example, Fragile X syndrome, where typically by the time a child was diagnosed, even though parents had concerns at a very early age, the average age of diagnosis was 3 years old, and that's even in the last few years where there's been more education about the condition. And oftentimes those families had one or more additional affected children before the first one was appropriately diagnosed. Shouldn't we expand the benefit of screening to benefiting the family versus the child only? And certainly that's brought up a lot of discussion. So what about BRCA mutations and other things that are autosomal dominant? So if you identify something in a child that's autosomal dominant, it could be a new mutation, but it's also very possible that one of the parents is affected. And so if you identify a risk of breast cancer in a parent of that child, isn't that a benefit to the child? And so shouldn't we be looking at it this way? And people certainly argue, well, if you screen for these things, adult onset conditions, and you tell the family, then you've taken away that child's right to not know that information. You take away the autonomy of the child. And what about testing children for carrier status? Should we be doing that when it's not really going to benefit them, especially at a young age, but it could benefit their parents? So with all these things in mind and a lot of discussion, the NIH put out an RFA for genomic sequencing and newborn screening disorders studies. This invited applications that proposed to explore the implications, challenges, and opportunities associated with possible use of genome sequencing in the newborn period. And 4 centers were funded, and the 3 other centers have shared slides and I'll just give you a little bit of a summary for each one. The group at Brigham and Women's and Boston Children's Hospital and folks at Harvard are studying 2 groups of patients. One are healthy newborns and their parents, and another group is from their intensive care nursery. And so they'll look at benefits, risks, and uptake and outcomes and in their cohorts. At Children's Mercy Hospital in Kansas City, Missouri, Stephen Kingsmore is really focusing on developing a more rapid turnaround time, because certainly we all know if this is ever going to be useful in newborn screening, you're going to have to do it quicker. And Stephen's had pretty impressive results in cases now, less than 72 hours that he's giving results back. And he's actually doing whole genome sequencing now. And he's focusing on infants in the neonatal intensive care unit also. So critically ill newborns, in order to qualify, they have to have had another type of genetic test ordered. And so they're randomizing their patients into ones that have sort of the standard genetic workup, and then those who go directly to genome sequencing. And then at UCSF, Bob Nussbaum was the PI there, but he's left to go to Invitae. He'll still be involved with it, but Jennifer Puck has taken over as PI at UCSF in California, where they're looking at stored dried blood spots and doing whole exome sequencing to see, what the quality of that is. And they have the diagnoses for the various conditions, but they're otherwise anonymized samples. And then they have another group of patients from Jennifer who have SCID conditions that have not yet been found to have specific diagnoses. So to look at the benefit of diagnosing those patients. And then our study that we call NC Nexus for North Carolina Newborn Exome Sequencing for Universal Screening, funded by NHGRI and NICHD. And our aims are to evaluate how next-gen sequencing can extend the utility of current newborn screening, to devise a clinically oriented framework for analysis for this, to develop best practices for incorporating it into clinical care of patients. All of the sites funded had to have 3 different components of their studies. So projects 1, 2, and 3, with each being of equal strength. And so project 1 is to generate a high quality whole exome sequence data set for next generation newborn screening. To implement an analytic strategy for what we call NGS, NBS, and incidental findings, and develop and evaluate novel bioinformatics approaches for utilizing in NGS, NBS. And we're working with the NBS TRN. All the sites are to come up with common data elements, and our data will be shared. It's a little bit like comparing apples and oranges, so it's been challenging to come up with common data elements, but we're finalizing that. Typically when you do whole exome, you come up with 100,000 variants, then those go down to 20,000 coding variants and 1,000 missense variants, and then the hundreds of truncating variants. with the vast majority being benign, and there's still no real gold standard for selecting variants. All of those that are thought to be clinically significant will be analyzed in our CLIA hospital molecular genetics laboratory before they're reported out. We currently have a workflow manager that helps us know if variants are likely benign, or are they VUSs, or are they likely significant, and certainly that helps a great deal. Our project 2 is looking at the clinical utility of whole exome sequencing. We'll look at whether it can improve the sensitivity by studying infants and children up to age 5 with known conditions like PKU and MCAD, and hearing loss, cystic fibrosis, and we'll see whether we're able to detect causative variations in the appropriate genes. We'll also look at whether WES can extend the range of current techniques. So can we screen for conditions that are not identifiable with current techniques? And then developing appropriate ethical and clinically oriented mechanisms to facilitate interpretation. We're using saliva swabs instead of getting an additional heel stick, and our pilot project has shown high quality DNA that came out quite well when we analyzed it for whole exome. Project 3, the LC component of this is, one part is binning genes, so that we've now, through various projects at UNC, looked at 500 different genes and scored them, and I'll go over the scoring. Our committee that does this is made up of genetic counselors, clinical geneticists, metabolic dietitians, people who formerly served on IRBs, and some of our students and residents also participate. We're developing decision aids to help parents decide whether or not they want to have their child participate and have their exome sequenced. What kind of information do they want? What kind of information are they interested in getting back in terms of conditions? And a modified scoring system has been devised by my co-PI Jonathan Berg and others at UNC that scores each gene depending on the severity of the condition, the likelihood if someone has a mutation in the gene that they'll develop the condition, or the penetrance, how effective are the interventions, what's the acceptability to that intervention, is it a special diet, is it an implanted defibrillator device, is it removing a stomach, things like that. So they all get scored. And then what's the evidence? So there are a lot of genes out there, a lot of the OMIM genes that really have little, if any, real evidence about whether that gene is even involved in the causation of a condition. So for example, with our scoring, I picked out PKU as an example. And so looking at the severity of the disease, it doesn't lead to death, but it certainly can lead to severe disability if untreated, so that got a score of 2. The 3 for likelihood of that outcome. The effectiveness of interventions got a 3, the highest score. The acceptability, well, it's not the easiest thing to be on a special diet, but it got a 2. And then the knowledge base, very good knowledge base for that, so it got a 3 and a total score of 13. And just to compare that to one of the other genes and disorders that we're looking at, if you think about multiple endocrine neoplasia, type 2B, this leads to early medullary thyroid carcinoma in children. Oftentimes by the time it's diagnosed, it's already metastasized, and it's 100% penetrant. Also, individuals with mutations in the gene develop pheochromocytomas 50% of the time. They also get mucosal neuromas and a marfanoid body habitus, although usually that's not obvious in newborns. So there is prevention. If you know a patient has this, you can remove their thyroid prophylactically. You can also do biochemical surveillance of catecholamines to pick up PHOs early. 50% of cases are de novo, so we're not going to have a family history. And the prevalence is, for all MEN2 types, estimated about 1 in 35,000. So certainly not very common, but probably in the same category as many of the conditions that we currently are screening for. The gene is certainly known. It's RET gene, and there's good genotype-phenotype correlation. Just in individuals showing the mucosal neuromas and the marfanoid habitus at an older age. And we know that certain mutations in the gene are associated with a very high risk, or level D, for developing thyroid cancer. So the recommendation is as soon as possible in the first year of life to remove the thyroid. There's no current population screening method available for that. So when we scored it, it got a 2 for severity of disease, possible death, likelihood of 3, effectiveness, effective interventions, acceptable interventions, kind of moderate, and good knowledge base. So it got the same score as for phenylalanine hydroxylase gene. So an age-based modified metric system has been devised to separate out the different categories of conditions. And so if you look at those with an earlier age of onset, infancy or childhood, and then the actionability score, how, can we intervene? So in the upper left would be what we would term our next generation newborn screening conditions, like those that are already on the RUSP and screened for, and then others like mutations in the RET gene and FAP, for example. And then in the upper right, adult onset, medically actionable conditions, things like BRCA1 and Lynch syndrome, and then those of childhood onset, but with fairly limited medical actionability, like Duchenne muscular dystrophy, for example, and others. And then the adult onset, non-medically actionable conditions in the lower right. So for the NGS, NBS, we would include those conditions that are currently on the RUSP, and then conditions that fit a similar profile to the RUSP. And we will have 2 different cohorts in our study. We'll have those with known diagnosed conditions and a healthy cohort. And those with known conditions will get the diagnostic results back. Those will include known pathogenic variants in the genes, as well as VUSs for that group, since we know what condition, what genes should be involved. And then both the healthy cohort group and the known disorders will get additional information about the genes that are in that NGS, NBS group. Then the groups will be randomized, and one section of each group will have the option of getting additional information, like for the early onset non-medically actionable conditions and the adult onset medically actionable conditions. And so those will include also carrier status for recessive conditions. Parents will have the option of that as well, learning that information. We are partnering with folks at RTI International, and they're developing online decision aid tools that hopefully will ultimately be publicly available to help parents decide whether they want to have this, what is whole exome sequencing, what kind of information might they get back. These are tools that help people participate in their health decisions. So it's a lot more than just going through, signing a consent, an informed consent document. We've finalized or completed a discrete choice experiment looking at 1,289 participants in terms of what kind of conditions are parents interested in getting, what are more important to them. And for example, if you tell them profile A, that there's a condition that 50% of children with the change in their gene will develop the health condition versus another condition where only 10% will develop the condition, and then give them additional information, like one is associated with mild to moderate physical disability, whereas the other one doesn't have any disability, and some have more options. So it's a way to kind of determine a lot of different things, not just, you know, this or that, but a lot of different variables. And then they're asked, like, which profile would be more important to you? And basically, the most important thing to parents was the likelihood of the occurrence. So they were a lot more focused on things that were, you know, 50% or more likely to happen versus those, you know, with like a 1 in 10% chance. This data is being further analyzed and will be published. As an example, like for Leber hereditary optic neuropathy, where about 50% of children are expected to have problems versus something like Rett syndrome, where, you know, the vast majority will develop problems if they have a significant variant in the MECP2 gene. And 23% said that profile A would be more important, but over 3/4 said that profile B, the Rett syndrome, would be more important for them to know about. And it's a technically challenging study. There's a lot of different steps to it. Parents will be surveyed. We'll see, you know, how they go through their decision-making choices and things like that. So can next-gen sequencing expand the utility of newborn screening? Well, it certainly will allow us to test for additional conditions. It will improve the specificity and sensitivity of standard screening. Molecular testing is already part of newborn screening for things like cystic fibrosis and hemoglobinopathies. It's also needed when, you know, you diagnose something like SCID or hearing loss, where you don't know what the underlying genetic cause is, if there is one. If you look at the different genes on the RUSP, you know, the majority have one or more genes that are known to be involved. Things like hypothyroidism, a fairly small percentage, but still a percentage, about 15 to 20%, are due to genetic causes. And so genetic testing can be helpful to further clarify the diagnoses. So certainly I'm not proposing that this will be a standalone test at any, you know, point in the near future. We still have a lot to learn about what's significant and what isn't. But already companies are utilizing panels, targeted next-gen sequencing panels for newborns to help expand newborn screening. And studies are coming out. Dr. Naylor and his group from Parabase reported their integrated screening models, and, you know, had quite a high sensitivity for detecting conditions that they you know, were blinded for in some cases, and you know, still were able to detect it through their next-gen sequencing, where they looked at a limited number of genes, I think about 160 or so genes that they looked at. And, you know, we know that we're going to have to deal with the ethical issues. If genetic sequence information is not returned, should it be stored? Where should this be stored? Whose responsibility is it going to be if we don't return information because we don't know that it's significant, because, you know, it's not known to be associated with an underlying disorder, but we learn that information, you know, or who's responsible, or is someone responsible for going back and recontacting families about that? What about parental rights to a child's DNA sequence if we're, you know, for people who argue against giving out some information, recontacting, And then already, as I said, some of the commercial companies are offering expanded newborn screening to patients. And if we do that, what about health disparities? Should it be mandatory or voluntary, let alone the demands on an otherwise already overly stressed public health system? And concerns as well about genetic discrimination. So I'd like to thank the other members of my team, Jonathan Berg, my PI, Don Bailey and Megan Lewis at RTI. Myra Roche is our genetic counselor, most closely involved with the study. Chris Rini in the School of Public Health, and Laura Milko, who's our study coordinator, and Kirk Wilhelmsen, who's helping with the informatics portion, and a lot of other people and people on our binning committee. Thanks.
You may have said this, and I missed it. Very interesting and adventurous experiments. I wonder, how long is your turnaround time? Do you know yet in terms of the whole exome? And did I get this? Are you going to be blinded to the newborn screening, the standard newborn screening outcomes of those kids as you do this?
For the diagnosed cohort, we'll know what their conditions are. And we won't be blinded.
Okay.
To it. We will be, I forgot what the other.
Yeah, the turnaround time on your
the turnaround,
exome.
Yeah. So we're estimating, we hope, like 3 to 4 months for our group. I mean, right now in one of our other studies, like Jim Evans had the NC Gene study as part of the CSER consortium group, and it's taking like 10 months or so. So, I mean, Stephen King's more.
How is he doing it?
I don't know how he does it.
Oh,
He's
come
got
on,
this
really?
Amazing pipeline. Yeah, one of the things I hope to learn more about is
Huh.
how he's doing it. Yeah.
Wow, fascinating. Okay, thanks.
Hi, I'm a consumer. I have a child with PKU, and I'm the chair of the Consumer Alliance here. Just a couple things. Thank you, first of all. It's fantastic. And a couple of things to think about. I'm going to ask that maybe you keep in mind as you move forward is, number one, what safeguards are in place, or can we watch safeguards or put safeguards in place that would ensure that insurance would continue to cover people in the future? And number two, maybe looking at ways to ensure that what we're currently screening for is covered, treatment is covered, because right now medical foods and medical formula are not always covered. That's it.
Yeah, and those are certainly very good points. Excellent points, and something that I think we all struggle with. And, you know, unfortunately, a lot of the others involved with this are not so closely involved with the follow-up of kids from, you know, who are identified with conditions through standard newborn screening. I think that's one of the good things about our center is we're highly involved, and we realize, you know, the limitations and the difficulty in, you know, funding long-term follow-up and you know, metabolic foods and things like that. So I know I struggle sometimes because this is sort of like, yeah, going, you know, into the future, and yet, you know, we're having enough struggles dealing with the present. So I appreciate your comments.